Let me just reiterate one fact that we talked about. NA is everything, right? NA, numerical aperture. So the higher the numerical aperture, I think I must have the one equation wrong or something. Somebody caught it who's good at math. Better than me, probably. Obviously better than me. That's why I'm in biology, because I'm not so good at math. That's why, I started in electrical engineering, and I got to like calculus physics, and I was like, wait a second, this is not for me. So I'm into biology. But anyway, the higher the numerical aperture, the better the resolution. The better the resolution is. Okay, so just keep that in mind. NA gives you better resolution. Okay. Okay, so second lecture. Principles and applications. I know it's the name of the course. And now this is the one. Hold on. Wait a second. Okay, so a lot of times you go to one of these workshops, I've been to a few of them, and the very last thing they'll do is like the last lecture usually, they'll talk about trends. Like, oh, this is the new thing, or this is the greatest new thing right now. Well, if you talk about it at the end, I'm going to go home, you're going to go home, and that's the end of it. You're not going to think about it anymore. So I figured, why not talk about it at the beginning? You might not know the techniques. We'll talk about some of these techniques as we go through, but at least you can start thinking about things that might catch your interest for your research projects, okay? So I'm sort of doing this out of order of a lot of microscope workshops, but I think at least it makes some sense. So I'm going to try to show you some of the latest things going on in the world of microscopes, and you can think about it over the next few days. And if you think they might be applicable to one of your research projects, then maybe you and I could talk a little bit about it and say, hey, yeah, it is. It might be useful for you for this particular application. Or I'd say, well, maybe not. Maybe this technique would be better for you. So definitely bring, I'm here for so many days. And, talk to me while you can. I'm going to give you my email and stuff when I go back. And I'm not, shy about helping anybody. I like helping people. So, just because I leave India doesn't mean, with the age of the internet, it's easy to get a hold of me. And I will help you, I consider you here my students in my class, so I always want to help my students, okay? Okay, so anyway, we're going to talk a little bit about trends here and how to avoid silly mistakes. Because like I said, I work in a core lab, and over the years, hundreds and hundreds of people have come to me and worked with me. And I've seen every mistake you can think of. Okay, I've seen some very, very good work. I've seen some very, very bad work. And I've seen everything in between. I've seen everything. A core lab sees everything. Like the first lecture, Dr. Say, he has his lab. So he doesn't see other labs so much. But I see all the labs. Okay? So I see a lot of different things. So I'm in a good position to point these mistakes out so you don't make them as well. Okay? So again, the other lectures from here on out are more technical techniques and things like that, but this is a really good lecture just to pay attention and try not to make some of these mistakes we'll talk about, okay? It will save you a world of pain, because I've made them myself, okay? I've been in research 25 years, I've made, hundreds of research mistakes, without a doubt. I still make them. Okay, we all do. Okay, anyway. Okay, so just a little bit about, I'm going to do this at the beginning, like I said. And feel free to ask questions about any of these techniques. I'm not going to dive into the techniques. I'm sort of going to present them. But if you have some questions as we go, just feel free to ask me. Okay. Let's start with something that's really obvious. Okay, I got my PhD in 1998. Okay. Most of you are working on your PhD now. Some of you have it. But I did my entire PhD in the world of film. I did a lot of microscopy. I took a lot of film, a lot of pictures. I spent a lot of time in the darkroom, okay? A lot. I was like what do you call it? Like a mole. You guys know what a mole is? Like lives in a cave. I was like in the dark all the time. Because all I did was sit in the dark room and take pictures, like fluorescence, like we talked about earlier. And then I had to go in the dark room. So I was just in the dark all the time. It was bad. I didn't see the sun. And then I lived in Ohio, so it was winter. You didn't see the sun anyway because their clouds were all gray. So it was bad. It was very bad. Okay. So film is dead. It's gone. When I started in sales in 2002, I probably had almost 1,000 customers. And at that point, I think I had like 4 or 5 people still use film. And then a couple years later, they were done. It was gone. Digital just became a thing. Why? Well, here's a picture from my PhD dissertation, okay? It looks pretty awful, okay? I mean, compared to what we do now. But basically, this was film. You know, we had to crop the pictures. We had to print them out. We had to make the little borders. Like, when we used to go to poster sessions across the country, we would literally have to have boards, like individual boards of our poster. So we'd have to make these physical boards with actual pictures on them. They used to sell little sticky letters and stuff. You could put the letter C or the letter D. You remember the days, I'm sure. It was crazy. So you'd go to your meeting, you'd have these boards of your poster. You get there, you'd stick them up on the thing. Now you have these really nice, you just roll the poster out and you're ready to go. You can do it all in PowerPoint or Photoshop or whatever, but back then we did not have that advantage. Okay, so this was a typical day when I was doing my PhD. I'd spend 2 hours taking pictures, one by one. I couldn't look at 2 colors at once in for us. I'd take like red, then I had to switch the thing, the filters, and I'd take blue, and I have to write my notes. This is blue, this is red, because you look at the pictures later, they're all black and white. You don't know what's what. Because like you said earlier, fluorescent, especially fluorescent pictures, they are black and white. I mean, we put color on using a computer, but they're black and white. And we'll get into this idea. He was talking about buckets and dumping photons in buckets. We'll get into that tomorrow. But it's basically a black and white technique. So then I have to dry and develop my film, another 20 minutes, make prints, like in the dark room, making prints. So a whole day, 6-7 hour day, I'd get 70 images. Okay. And we have cameras now, like cameras I use, I could take, 50 frames a second easily. There's a camera I used to sell, it was called a red light camera. It took 111,000 frames a second. So you take 111,000 pictures in 1 second. Okay? Like that super stop time lapse kind of picture, like you could see like a bullet coming out of a gun, and you could see the bullet like spinning because it took so many frames a second. It stopped time. Okay? But just, like I said, I got my PhD in 1998, not forever and ever ago, but what's that, 19 years ago. And while I was working, I was doing this kind of stuff, okay? So 6-7 hours for 70 images. Okay, so I think all of us that have been doing research for a long time would say everything's gotten faster. I mean, it's gotten ridiculous. Like, my first postdoc was building a knockout mouse. Okay, and I don't know if you guys know what's involved in knockout mouse, but you have to know the sequence of the gene in the mouse, and you have to know the regions outside it. So I spent months and months just figuring out the sequence of the gene I was working on and the sequences around it, because we used to pour big, like this big radioactive gels, and we had to load samples. I mean, it was painful. It was really hard to do, but it took a lot of time. But now, these days, you guys are lucky, you have all this extra information. You can just look up the sequence now and start building knockout mouse like that. The sequences are all there because all these advances in technology. So I've sat down and thought about my whole PhD, which took me almost 6 years. I switched labs too. But anyway, I think I could do the whole thing in like 5 months, maybe 6 months, a little bit of luck and a little bit of whatever. But the actual work, I think could be speeded up that significantly because of the technology advances. Okay, so getting back to this idea of trends. So, is that bad? So should I say yes or no? Want to say yes? Who says yes? Who says no? What do you think? Yes?
No.
No?
Say no.
You pick. Okay, that was a wise choice. Okay, thank you. Okay. So what they want most of the time, they'll come and go, oh, I need all these pictures. I need lots and lots of pictures, and I need them now. I need them like yesterday. I need them very quickly, okay? And that's fine. We have the technology to do it. But the thing is, the quality suffers a lot of times. People want all this stuff. Okay, so if I take 10,000 pictures today, let's say. I mean, there's days I take way more than that. Do you have time to look at 10,000 pictures? I mean, do you really? You don't. I mean, it's sort of silly in a way. Why are we doing all this? Why are we using all this computer space? Why are we taking so many pictures when, I mean, who has time to sit there and look at them? It's just, it's sort of silly. But I get these, my boss wants, or whatever, okay? It's usually the boss wants something. And then I have to go talk to the boss and say, well, you're sort of overkill here, so let's not do that. Okay, so the big thing is, I mean, research is not easy, okay, in general. It's difficult because, I don't know if you have an Indy, but you have textbooks, and then like you have questions at the end of the chapter in the textbook. Okay, and if you answer those questions, you look in the back of the book, and it tells you the answers. You have those kind of books, correct? Okay, we have them too, right? Well, research doesn't work that way. You can't like look at the answer. You can't do that. You don't. It's research because you don't know the answer. Okay, so it makes it difficult. I mean, research is not easy. That's why you go through all this schooling. That's why you go to these workshops like this. That's why you study hard and do your work and do your experiments, because it's not easy. And you have to prove that you're right, because chances are what you're working on, nobody knows the answer. You have to come up with the answer. So it's not easy. So the thing in America I see right now, and I'm sure it's here to some extent too, is we have a lot of pressure where we're at to get the money. Like the grant money is driving everything. Like, I mean, I know a lot of labs where I work, well, they didn't get the grant, so everybody gets fired and they're looking for jobs, and the PI now is working in somebody else's lab trying to get a job. I mean, it's a mess. It's a big mess. The other one is, and most of you students here, you're trying to graduate, right? Well, if you keep doing experiments, they don't work, it becomes very frustrating, and you just want to graduate. So it becomes, I think, critical to sit and think about it. Like in the old days, I mean, not, I'm not super old, but older, like I was talking about, we really thought about the experiments because they were so technically challenging to actually do them. You were thinking to yourself, do I really need to do this? Because I don't really want to, but if I have to, I'll do it. But now, when the age, like, I don't know how much you see it here in India, but kits and like little kits to make the experiments easier, to do PCR and to do Western blot stuff. We have all these kits in America that are fast, and you add reagent A, reagent B, reagent C, and it does something and gives you a product. Well, the problem is nobody understands anymore because they just do, well, what did you do wrong? Well, I don't know, I put A, B, and C. Well, what does A do? What does B do? What does C do? They don't know because it's a kit. They're just so used to doing these and not thinking it through, whereas in the older days, we had to think it through because we didn't have the ability to use these kits, okay? So I guess what it comes down to is think about what you're doing. Don't do experiments that are going nowhere, okay? They always say the best scientist, at least, the whole idea is to publish papers, right? I mean, that's really what it comes down to. So I've only met one person that could do this in all my years. So they always say, think about what question you're trying to ask, and think about what that figure in a paper will look like. And do the experiments accordingly to try to generate that figure, okay? And like I said, I only met one guy, and everybody's like, he never comes to work. He's never at work. He's always slacking off or doing something. And the thing was, though, this was the only guy I ever met. He could literally think like that. He could plan experiments that would make, figure 1 in the paper, figure 2 in the paper, figure 3 in the paper. He was so efficient. It was ridiculous. I never met anybody like him before. So think what you're doing. So we have a saying that says a stitch in time saves 9. Does anybody know what that means? A stitch in time saves 9. I don't know if it's we have a guy in America, Ben Franklin from back there. I'm not sure if it's one of his sayings, but he had a lot of these kind of sayings. So what that means, basically, is like, let's say you have clothes, right? Everybody here's got really nice clothes on, and you get a little rip. Well, if you take the time to sew that rip and stop it from becoming a bigger rip, then you save time. Because if you wait, it's just going to get bigger and bigger and bigger, and you have to spend more time sewing. So a stitch in time saves time. Now, do you have any sayings here like that? Something like that here? Like, anybody? Anybody got something like that? No? But a lot of different countries have sayings like this about saving time or doing things more efficiently. Okay. So I was going to ask if you got one here. See my first animation there? I was getting better at making these. Okay. So basically what it means is, if you take your time, you'll prevent more work later on. Okay? So science is a lot like that. You want to take your time, do it right the first time. You don't want to do it wrong, and do it wrong, and do it wrong, and do it wrong, and then finally sit down, think about it, and go, oh, I should have done it this way. Okay? Okay, so I see these mistakes constantly. Okay? Over. Okay, the first one is, we'll get into fluorescence imaging here soon, but people come to my lab, and they come over, and they go, I need to look at these cells. I'll say, okay. And I'll say, what color? They'll go, red. I go, okay, there's about 5,000 different kinds of red now. So what red? And they'll tell me some dye or something. Like, I've never heard of that. So I'll get out my books and look it up, or look it up on the computer, and I'll say, I can't look at that. And they'll go, what do you mean? I said, I do not have the capabilities here with my microscope, the way it's set up, to look at that dye. And they look at me like I'm crazy or something. So we'll get into tomorrow about like different wavelengths of light and how fluorescence works. But always check, if you're going to use a particular microscope, make sure that microscope can see the dye you want to use. Okay? Because if not, you're just wasting your time. Okay? Fixative. We talked a little bit about this when we were talking about making slides, right? So fixative, a lot of the fixatives are paraformaldehyde or formaldehyde. And what happens is, if they go bad, like if they sit a long time, they basically turn into methanol, a lot of them. And they'll hurt your cells. They won't help anything. They'll destroy what you're trying to stain. So people think they're doing good. Well, I took my piece of tissue out of the animal or human sample or biopsy or whatever. I put it in the fixative. Well, how old was the fixative? Well, I don't know. I made it like 6 months ago. And it's like, wait a second. So my first, like when I started in my PhD lab, not my first advisor, but my second advisor, she took me aside and she said, you're going to do a lot of microscope work. And you're going to make a lot of slides. And she goes, you're going to make up your fix every single day fresh. Every day. And it takes like 15, 20 minutes. But she goes, you're going to do that every day. Because if you do that, you will avoid a world of problems. And, she had done this for years, and I took her advice, and I really did not have the problems that I see now with a lot of labs. And sometimes it'll take us, quite a while to figure out, oh, the fix was old, it was just sitting on a shelf. Or somebody else made it a long time ago. I mean, part of it's, you're lazy, you don't want to do it. You're tired, you got other stuff to do. You got to study, you got a different experiment to do. But this fixative idea of making it fresh, I'm not saying you have to make it every day, but a lot of times, I wouldn't go more than a week. Make it up. I mean, if there's any question in your mind, like, oh, it might not work because it's old, make a fresh batch, okay? It just crushes your experiments. Okay, so I get lots and lots of people. Do you guys have those 96 well plates with the little circles? Okay. They're nice for doing ELISA and other techniques like that. They're the worst thing in the world for a microscope, okay? They are just totally awful, because they're these little tiny wells, the light bounces all over, they're plastic, which is bad, and they're horrible. And people come over and they're like, well, can't you take a better picture? It's like, can't you bring me something better to start with? I mean, it's just bad. 96 well plates are for ELISAs. They're not for imaging. I mean, I know you have the cells in there or something, but don't use them unless you absolutely have to. Avoid those at all costs. Okay? Okay, now, do you guys have a flow cytometer here? No? Okay, so flow cytometry is, you take cells, they're in liquid, and you can stain them like you stain for a microscope. And what it does is it passes a detector, And the detector can either take a picture of that cell, or it can decide, okay, positive, negative. Like if it's green, it's positive, it goes this way. If it's red, it's negative, it goes this way. So it can sort too, a flow cytometer. So it's almost like coming out a hose. So each cell's coming out a hose, and then it gets to the, like a detector on the hose and decides, oh, it's red, it's green, whatever, this way or that way. Okay. Now, flow cytometry is used a lot for immunology cells, because they want to look at all these different kind of markers. Like they want to look at like different things on a T cell or a B cell or a macrophage. So sometimes they'll look at 4 or 5 different markers and say, okay, this one's blue, and then we sort it again, now it's blue and red. So they have a lot of dyes for flow cytometry. Lots, I mean hundreds and hundreds and hundreds. And where I work, a lot of people do flow cytometry. But the dyes are crazy dyes. They're not microscope dyes. So if you see a dye that says, oh, keep it farther away. Is that better? Okay, perfect. So if you see a dye that's used for flow cytometry, chances are it won't work in a microscope, because they have these very strange, like when we look into fluorescence, excitation and emission properties that are not designed for the microscope. Plus the other thing is, think about it, it's coming out of a hose, and a lot of times it just needs looked at for like a half a millisecond, like it's green, it's blue. So the dyes can't handle light hitting them either. They tend to what we call bleach, like the fluorescence wears out, it won't work anymore. So flow cytometry dyes are made in such a way that they only have to last for maybe a couple seconds, which is fine. But if you're trying to use it on a microscope, you're looking at the slide for maybe a minute, 2 minutes. You need a different kind of dye that can handle that fluorescent light, which is high energy, hitting it. And it's hurting it. I mean, fluorescent light is nasty. I don't know if you've ever, probably like you're driving down the street at night or something, somebody hits you with the bright lights from their car. Well, multiply that by hundreds and hundreds of times. That's fluorescent light. Like, sometimes I'll be working on a microscope, I have to take the light off to see if it's working. I mean, it hurts your eyes. It's like, wow, it's really bright. So that light is damaging. So don't use flow dyes, okay, for a microscope. And like I said, it goes back to rule number 1. Go check. You've got your microscope person here who runs it. Before you start your experiment, just go down and say, hey, I want to do this experiment. Can I use this dye? I mean, better than doing the whole experiment, walking down there and then have them go, no, you can't. Okay, so again, stitching time saves money. Okay, we're going to get into this a little bit in the next couple of days when I start talking about antibodies. But this whole idea of immunohistochemistry or immunofluorescence. Immunohistochemistry is basically you're using enzymes on your antibodies to make like a brown product on a slide to detect something. So that's what that is. Immunofluorescence is what we're going to talk about, using fluorescent molecules to look at things on a cell or a slide or whatever. So when you look at an antibody catalog, like let's say your boss says, I want you to study the protein Rho, okay, like a signaling protein called Rho. So you look at the catalog and there's all these antibodies against Rho. Well, you find this one and it says, it works for a Western blot, but it doesn't work for immunofluorescence. Well, you're going to do immunofluorescence. So try to find proteins, and we'll look at some antibody catalog pages to show you, hey, make sure that the antibody you're using does that, will work for this application, okay? Because just because an antibody recognizes Rho on a Western blot does not mean it's going to recognize Rho in immunofluorescence. Now, why is that? On a Western blot, you've denatured the protein. So the critical thing about a protein is it's three-dimensional structure. Like, it's got to have that certain three-dimensional structure in the native form. The conformation has to be in such a way for the antibody to recognize it. Well, in a Western blot, you're running on a gel, you're denaturing, so you're opening up the protein like this. So you've lost a lot of that three-dimensional structure. So some antibodies will recognize that, other ones won't. Okay? So I get this a lot. Well, they're like, it worked on a Western, and we can't get it to work here. I said, well, it's a different world. This is a different world. The other thing, too, is if you're going to work on a certain project, like a certain protein, it's worth it to go back to the literature. We have all this internet now. Look in the literature. See if somebody has done something similar to what you're trying to do. Because if they've already done it, Like if they've done the exact same thing, you can't publish that because they've already done that. Okay, so that's one reason. The other reason is if you're doing something similar, find out what antibody or what they did. Look in the literature. I mean, it's your best friend because it's this whole giant mass of scientific knowledge that's right there at your disposal. You don't have to do any work, just get on the internet and look. And you can find out what they used. So why go to a catalog and look at 27 antibodies and say, well, maybe this one worked or maybe that one worked. Go to the literature. This lab is doing something very similar to what you did. Use what they did. I mean, it only makes sense. Think ahead. Think ahead. Don't just bash your head on the wall for no reason, okay? Think ahead. Okay. Okay, now this one, number 6, is the worst. So somebody will call me up on the phone and go, can I come over and work with you like on Thursday at 2 o'clock? I'll say, sure, that's fine. What do you want to do? Oh, we're looking at this protein or something. Or we're trying to see if we add this drug, if this protein becomes more expressed or less expressed, something like that. So they come over, they bring 60 plates, 60 dishes, or 60 slides. And I'm like, okay, what are we doing? They sort of explain it to me. I said, okay, hand me your first slide. Hand me your first slide. I put it up. I sit there and look at it, and I go, didn't work. They go, what do you mean it didn't work? I said, it didn't work. I go, let me look at another slide. I said, it didn't work. Didn't work. So I see this all the time. They do every condition, every part of the experiment. They do the whole thing. Like their boss said, I want you to answer this question, this question, this question, this question. They do all of it. But the problem is, they never stopped and made sure it worked first. So they never did the positive controls and the negative controls to make sure the experiment worked. So you have to do the positive and negative controls first to make sure everything works. Because like I said, research is difficult. Research is hard. Do the positive and negative controls. Once you have that all figured out, then it's time to do the real experiments. Because I have people that are really good scientists. They'll come over to my lab and they'll take 2 or 3 weeks to get everything working just right. Once they spent that 2 or 3 weeks troubleshooting, figuring out dilutions, figuring out concentrations, figuring out how to do it, then they'll do the real experiments. But they'll spend 2 or 3 weeks just making sure the system works. You have to make sure the system works first before you do all of it. So I always call that amateur hour. You know what amateurs are? They're not professionals. Okay, a professional person makes the experiment work and then does the actual experiment. Okay, so it's make it work first. Because if not, you're just wasting your time. Okay, I get this one all the time too. So they'll come over with brain, and they'll say, I'm looking for, SARC or something, some protein. I go, okay, let's start looking. Did you do your positive controls, negative controls? Yeah. So we'll start looking, and I go, I'm not seeing anything. Well, the literature says that this antibody should work. Okay. And, we'll look and look, and I was like, it didn't work. And then I'll ask them, are you sure that this protein's in brain? And a lot of times they'll look at me and go, no. Well, why are you doing this? Well, my boss wanted to see if it's in brain. I said, that's fine. But now you've brought me a slide. You don't know why. We don't know why it didn't work. We don't even know if that protein's in brain. So how are we supposed to figure out? Did the staining not work because it didn't work? Or did it not work because that protein's not even there? Okay, you see the problem there, right? I mean, you've got to make sure what you're looking for is there. So look in the literature, make sure something's there. And a lot of times this is real common. They want to look at immune cells like T cells or B cells or macrophages. They want to look at tissue. You used to do some of that, right? You're like, Tom does a lot of that. Tom does lots of that stuff. Okay, so if you want to look at T cells, and you're trying to look at, there might be in the skin or in the lung, there might be very, very, very few T cells there in the system you're using. Well, so they bring it over and it's like, I can't tell if it worked, I don't really see any cells. Is it because there is none? So why don't you just get some thymus from the mouse? The thymus is a big organ in the mouse here that's just full of T cells. There's millions of T cells. Why don't we take that? Why don't we use that and stain that with our antibody and make sure it works first? Okay, so we're not wasting our time. Okay, so think, find a protein when you're going to use a new antibody and your boss has you looking for something that you're not sure if it's there. Find something that'll be a positive control for you, that you know, hey, my staining worked here, like in the thymus, but I can't find that in the brain. Well, so what that tells you then is it tells you, hey, maybe that protein is not in the brain, okay? So, think. It's all about thinking. It's not so much about doing, it's about thinking. Okay. We'll get into this eventually, secondary antibodies. So, there's hundreds and hundreds and hundreds of different kinds of secondary antibodies. But the one problem is I'll get antibodies, like, they'll have a primary antibody that's made in mouse. So then they come in with the secondary. We'll get into this more in a couple days. But it'll recognize that. Well, the problem is this is made in mouse. It recognizes mouse IgG, which is on mouse immunity cells. And then you're using this setup to recognize this antibody, an IgG, made in mouse, and you're using a secondary that recognizes mouse IgG. Well, the problem is they use it on mouse tissue. Well, mouse tissue has IgG. It has its own IgG from its immune cells. So I'll get this where you see it just sticking all over the place. So they're using a secondary antibody that's cross-reacting with their tissue. This is just wrong. They just didn't think it through, okay? So again, think about it. Okay, another thing we see a lot of now, I see a lot of it just because I work with so many labs, is just because it's published, okay, we hope it's true, we believe it's true, we go through peer review, we hope it's true, but don't always believe it, okay? Because a lot of times I have people come in and say, well, we did this because this lab did it, but we just can't get it to work, and we'll do it again, and again, and again, and again, and again, and again. So I worked with the lab, like probably one of the best diabetes labs in the world, and there was these studies that came out about how the cells in the eye died in a certain manner, and it was from this one lab, and they were showing like 20% of the cells in the retina were dying. So we did the same experiments. We couldn't show anything. And we had another lab in the building, did the same experiments. We couldn't show anything. So we did it again, the other lab did it again, couldn't get any work. So what it turned out was that particular lab that got it to work somehow fed these mice this totally different kind of diet that let them get those kind of results. And the rest of us just couldn't do it. We couldn't make it work. But this took months to figure out what was wrong. So, and really what it was is that the diet they were using was sort of, they shouldn't have been doing it probably. But like I said, just because you see it published, don't always assume it's true. And especially if you try to replicate, you can't. Eventually you gotta start believing yourself, okay? Okay, eventually tomorrow we'll get into different kinds of images and different kinds of extensions on images. But like I talked about, I tried to bring up at that earlier lecture, was how there are pictures that are art and there are pictures that are science, okay? And we'll talk about this more tomorrow. But by the time this course is done, you'll know, don't bring me a JPEG image, like .jpeg for image analysis, okay? JPEGs are for PowerPoint and art, okay? Your phones take JPEG images for the most part. You see them as JPEG images. They're nice, they're pretty, they look good. But as we'll get into it, I'll explain it a little bit more, they're not for measuring. They're not for detecting. Like everything that Dr. Sanai showed you this morning, basically all those are TIFFs. TIFF is the way you want to save images for science. Okay, always TIFFs, and we'll talk about that more, like I said. Okay, the other big thing is that people just don't do it, I don't know why. When I first started in science, I didn't do it, but you're at a university, right? You got a lot of smart people here, okay? Some are in your department, some are in your lab, some are in another building, and you've got other universities too, in India and across the world. Well, if you don't know how to do something, ask around. Don't just do it and try to do it. When you realize to yourself, I really don't know what I'm doing. Try to find somebody in another lab that's done it or that can help you, okay? Because science is a team concept now. It's not just somebody working in a corner all by themselves anymore. It's a worldwide undertaking, and you've got to be able to take advantage of other people and the resources that are available to you. So what I have for myself personally is I have what I call a 5-minute rule. Okay, so my 5-minute rule is like, say with PowerPoint, I'll sit there and I'll try really, really hard to do something. Okay, and I'll try, and I'll try, and I'll try, and I'll try to look it up on the internet, and I'll try to figure out how to do it. But when 5 minutes is done, I'm done. I will not sit there and continue to waste my time trying to do something over and over. I will go find somebody that knows how to do it. Okay, so that gets back to the point of you're in science. It's a group effort. You need to start making friends and building a network. It's a network job. It's a team effort now. So if you know somebody in some lab that's really good at a technique, hey, keep that in mind. If you have to use that technique, go talk to them. Okay, it's a team game. It's a team effort. So I have a huge network in Cleveland, Case, and through the imaging world. Part of it is I like to talk. Okay, so I've made a lot of friends. But, you know, I mean, here, I'm still in contact with Srijit, right? I still talk to you. And Manish is coming tomorrow. I still, you know, keep in touch with him, Sivan. So people I worked with years ago. When I was in sales, I still talk to those people once in a while. When a problem comes up in my lab, I mean, I might not talk to a guy in 10 years. I call up and say, hey, I know that guy knows the answer. Why would I waste my time trying to figure it out when I know this guy's an expert? So it's all about saving time and not wasting your time. Okay, so the most important thing when you do microscopy, making slides or your cells or whatever, it's all about the sample prep. Because people bring me stuff all the time. We put it on a microscope, I take a picture, and they go, that looks awful. That's just horrible. That is just unacceptable. I said, I'm just taking a picture of what you gave me. I mean, it's not me. This isn't me. This is not my fault. This is your fault. You did a lousy prep. Take your time. Be gentle. You know, they're cells. They're fragile. Tissue, it's fragile. Be gentle, okay? Be gentle to these things. Because if you bring me garbage, I'll give you back garbage. Okay? You bring me something good, I'll give you something good. Right? It's basically what it comes down to. Okay. Trends. So, optical microscopy is so big now, it's its own, I guess you could say it's its own discipline. It really has gotten ridiculously big. I have a very hard time keeping up with it, the newest stuff and whatever. So I'm just going to show you a couple things that if you want to, you know, get into it and think about it, these are sort of easier ways to stay up on the latest stuff and the latest kind of news, and it reviews old techniques and things like that too. So there's actually a magazine called Biophotonics, and basically what it is, it's about microscopy in terms of biology. Okay, there's a lot of ads in it, and a lot of companies, you know, put ads in there selling different microscope products. But there's nothing wrong with companies either. I mean, companies are all in it with us too. So a lot of people, companies are very knowledgeable. But there's a lot of ads in there, a lot of good articles about, you know, this new technique or that new technique, or, you know, this kind of microscopy or doing this. It's just and it's a well written. It's not very big. You can get it online, and I think one of the best things about it is free. Okay, it doesn't cost you anything. Okay, I used to have a big stack of these in my office because I've been getting it for years. Okay, it's a free thing. It's sort of nice. You know, it's just something if you want to read microscopes. They're not super technical articles. They're written more towards, like, you know, I'd say not quite beginners, but like intermediate kind of people. So it's a good thing to keep up with just to see what's going on now. Okay, now I think a lot of science now is changed because of the internet. Okay, I'm on this thing called Confocal Listserv. So what Confocal Listserv is it's basically all the confocal people in the world. We're all on this, like, a list server. And what it is, somebody will say, hey, like somebody in France will say, I'm having trouble with this on this confocal. And then, you know, a couple hours later, 3 hours later, here comes an answer from Australia, here comes an answer from Canada, here comes an answer from India. I mean, you know, it's great. I mean, you know, the whole world's in with you on this. It's really, really nice. And it not only has, like, academic people on it, it has industrial people. So I'd recommend, if you're going to do lots of confocal, get on this thing. Because lots of topics come up that are interesting, and you have this whole group of professional scientists that are microscope professionals that will help you. I mean, some people just love to answer stuff on here. So like I said, why waste your time, days and days of time, trying to figure something out? You've got this group of international experts that will help you. I mean, it's silly to waste your time. Okay, so like I said, if you're really going to get into this is a great thing to be on. I'm sure there's these for other areas in science too, maybe your specific area. I don't know much about this, I can't comment on that, but I would think there is. I mean, everything's become very global, so I would think that's out there. Okay, there's some rules. You can't post, you can't just try to sell things on there. It's not made for that. It's not just for selling. And it keeps it from, just turning into a sales thing for the salesman. I mean, the salesman will offer technical advice, which is nice. The other nice thing here is a lot of workshops are listed. So if you like this workshop here, and let's say you want to specialize in something, then it'll show you, like a lot of people advertise their workshop, like we're having a workshop in, wherever, Maine or something about this or that, or, France or whatever. So a lot of international stuff's on there about workshops. Okay. The other thing is, it's a good place for jobs. Like, microscope world is very nichey. There's only, I wouldn't think there's, maybe 2,000-3,000 actual microscope people in the world, but the jobs are posted on here a lot. So pretty much everybody does a lot of confocals on this list. Okay, a good place to look for microscope-based jobs and postdocs, stuff like that. Okay, here's what it looks like. Like, this is my typical email that I'll come in the morning. So I'm looking at my Confocal Listserv, but alignment protocols for this particular confocal. I think yours is SPA. Live imaging discussions. An announcement here for a certain course, another course, a Zeiss career opportunity. I mean, it's just a really good place to go for microscope stuff, sales, knowledge. If you have a question, I put questions out there, and I've gotten answers like a couple hours from all over the world. It's great. Okay, so let's talk a little bit about what's the latest, greatest kind of trends now. Okay, so the biggest thing right now, hands down, in science, at least where I'm at, and I think most of the world right now, is probably CRISPR. Do you guys know about CRISPR? CRISPR gene, right? Like being able to basically go in and cut out a piece of gene, put the new in, but not like in the old days, like in the, cloning a piece of tissue in a test tube, but basically being able to fix a genetic defect in an organ or a whole body at once. Okay, it is smoking hot. It is the hottest thing going right now. Almost every lab where I work is jumping on this to some extent or another. This is the big thing. This is the really big thing coming. When I was working on my PhD back in the 90s, apoptosis became the big thing. It was the big fad. Like, I remember going to Cell Biology in 1996, and like every other poster was apoptosis, 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 apoptosis. So science comes in fads too. Like, I think, fashion has fads too, I'm sure, right? Like this kind of dressing like this is in, or dressing like that is out. But science has fads too. And right now the big fad is CRISPR. So if you have any interest in this, or if your lab does something like this, it's a good place to be right now. Okay, CRISPR's the hot thing. Stem cells right now is also big. I work with many labs that do stem cell work. It's amazing, the stuff they bring me. So I work at the lab down the hall for me. They'll take my blood, because I have nice big veins. Everybody likes me for blood, which, I don't know, they just like me because I have nice big veins. So I'll give them blood. They'll take my stem cells out of my blood. They'll come see me like 115, 120 days later, okay? They will bring me stuff in a dish that looks just like retina. I mean, it looks just like retina. If you didn't know any better, you would think they took it out of a mouse or out of a person, because they're able to grow retina from the stem cells in my blood. Another lab I have does the same thing. They work with pancreas. They grow pancreas from the stem cells. I mean, it's just unbelievable stuff that's going on right now. Now, it hasn't quite gotten to cures and things for people, but it's coming. There's little bits of the staff, but the clinical trials will be coming. These are the hot new things. Now, I know it's not microscopy, but it's hard not to, if we're talking about trends, it's hard not to talk about the two biggest trends I think going that I see all the time, just so you're aware of them. Okay. So I'm guessing you've heard stem cell work here is big too? Like different labs in India, I'm assuming? Right, it's big. I mean, it's going to be a very big thing. Because when I started my PhD, gene therapy was the big thing, being able to fix genes one at a time. Like if you had cystic fibrosis, which is a mutation in the lung, they were talking about using a cold virus or something to like infect the lung and alter that DNA and fix it. Well, it never really worked out, which I think is one of the problems I never got jobs as a microbiologist. But these trends come and go, but right now these are the two hot ones. Okay. So let's get more specific about what's hot in optical microscopy, since this is an optical microscopy course. Okay. Whoops. Super resolution is the hot thing. Okay, so what super resolution is? I think I got a slide on it. Okay, so what it is like when I first started working with microscopes, you were taught that the physics says, like when we talked about how important resolution is, like the ability to differentiate two objects from each other. Well, basically we were told if you use the physics and use the formulas and stuff, then maybe you could go down to like, you know, maybe 300 nanometers is the best you could resolve with an optical microscope system. Well, we'll talk about this more in the next couple days, but they came up with these tricks that actually sort of cheat physics in a way that let you get better resolution than this, okay? And this is the hot thing at work. We have I don't run it, but in our, over in our genetics department, we have what's called a STED. This one here is STED. And we can get down to resolution like 20 and 25 nanometer resolution, which is, you know, some years ago, I mean, the physics said you couldn't do it, so you figured you'd never be able to do it, but some very smart individuals figured out how to do it and got around it. So they got around that theoretical limit of resolution. So that's really, really hot. Okay, now this one's been around a little bit longer, but another technique is called TURF, okay? TURF is a technique that lets you look at cells, but only very, very, very close where they touch the cover slip. So TURF only works like, I think maybe around 50 micrometers from the cover slip, maybe 100, yeah, around 100 maybe. But it's made for like cell adhesion molecules, molecules that stick cells down and interact with the matrix or something like that. So it's a very niche-y kind of technique, but when it works, it's beautiful. So like here on the left is just a wide field microscope picture of what's called focal adhesion kinase, which is a molecule that helps cells, like part of a signaling complex, helps cells stick down. And then if you use TURF, this would be the exact same kind of sample, but you get this just totally different look, okay? So this is pretty hot too, but it's very niche-y. You've got to be working on basically cell membrane proteins, but this is sort of hot too. I've seen quite a bit of this lately. Okay, the other big thing, when I first saw this, I really freaked out, okay? I went to a demonstration. What this is like if you look at a microscope, you know, you're gonna look at a tiny piece of tissue, right? And you take that picture, you have this picture of just a tiny bit of the whole piece of tissue. Well, people wanna see the entire, this looks like brain, I guess. So this is probably a big section that's something you probably see on a slide is probably as big as my fingernail, like a part of a mouse brain. So it's a big thing. If you took a microscope picture, you'd have one little tiny area, you couldn't see the whole brain. So what they did is these companies came up with ways to take higher magnification pictures. But if you could see it, I left the background up, but see how it's like 1, 2, 3, 4, 5, 6, it's multiple pictures. So it takes multiple pictures at a high mag, stitches them all together. So we call this either scan slide or stitch slides. And it takes these multiple pictures, puts it all together as one big picture. So it's that you can use higher magnification, but you can get the picture of the whole piece of tissue too all at once. You get the finer detail, the higher magnification, and you get the bigger overview picture too. Okay. So the reason this freaked me out is I went to a demonstration and the guy goes, I'd like to show you this new machine. So he took like 100 slides, 100 of these H&E slides like I showed you. He put them in this holder. Okay, like a holder like this. And he basically pushed a button. And we sat there and watched it. And it basically puts the slide on the thing, finds the tissue, takes all these pictures, puts the tissue sections together just like this. And I mean, it did. So you could just put it on, walk away, and do hundreds of slides. So I'm thinking, how am I going to have a job? I mean, it's just crazy how nice it is. But the thing is, the reason I still have a job is this doesn't answer all your questions. I mean, it's great for just, being able to look at a whole bunch of H&E slides. If you want to look at a whole bunch of different conditions, you don't have to sit down on a microscope and do all the work. Like, you don't have to use my skill set to do it. And it doesn't work so well for fluorescence. But this is the heart and soul of digital pathology, okay? Now, digital pathology, pathology is the study of disease, right? So, the one thing I don't know if you know about India is that you've killed a whole bunch of American jobs for doctors. Did you know that? Because we used to have a lot of radiology positions, people that read x-rays. So there was whole residency programs in America that all they did was they taught the doctors how to read x-rays, right? But now it's much cheaper. They take the x-rays, they digitize them, they send them to India, and your doctors there in India read them, they send the reports back. So literally dozens and dozens and dozens of medical schools in the United States don't even offer that anymore. It's not even a residency at a lot of hospitals anymore. And it's all, from what I've heard, it's all been shipped to India basically. So, but that's radiology. But pathology is the same idea, where doctors look at the slides, the microscope level, and they do the same kind of thing. They can now ship these all over the world, have a pathologist look at it, look at these sections generated by this, and just make the diagnosis off that and send the report back. Okay, so this kind of stuff is the heart and soul of digital pathology. We had one of these at Case, it broke. They're writing a grant for another one. I think you're talking $180,000, $90,000 range, something like that, maybe $200,000. The prices come way down. Okay. So image stream. So image stream, you could like later on, like you guys can just click on this link. It's like a little movie about it. But basically what it is this like this idea of flow cytometry. Remember, like I told you, the cells are coming through a hose, like one by one. But image stream will actually take a picture of all those cells. So you can have thousands and thousands of cells come through with a fluorescent tag, and this image stream will take pictures of that, one by one by one by one by one, which is nice. The only thing is it's not like cells on a slide where they're spread out, maybe interacting with something. These are just loose cells, But if that's the application, then that'll work for you. Okay, laser tweezers. Okay, laser tweezers. Let's take a look at this website if we can. Now this I might need help with too, but I'll try to. So I think you right click on the link, right? Or am I wrong? You just click on it. Oh, okay. So maybe, the computer's probably not on the internet. But anyway, okay. But laser, should we try again? I don't know. No. Okay. But anyway, laser tweezers are basically use an optical light to push things around on a slide or in a system. Like you have cells, you can literally push them and move them and manipulate them with these like optical traps, okay, and move them around on the slide. I've been seeing quite a bit of that too lately at different institutions. Okay, now this one, image fusion of mass spectrometry and microscopy and tissue mapping. Okay, so do you guys, I think you guys have, or getting, I thought I saw on the way a mass spectrometer. It's coming, okay. So, because I thought I saw a sign or something about it here. So what mass spectrometry is it basically looks at the chemistry, the chemical nature of a given sample. And it's able to very accurately, based on a molecular weight, determine what that particular molecule is, okay? Because the molecular weights you're talking like, like 13,426.22. So if it comes out at that molecular weight, it's probably that molecule, okay? So very powerful technique to determine what a sample's made of, okay? So somebody got really clever, this guy out of Vanderbilt University, combined this idea of being able to tell what something's made of with microscopy. Okay, and it's basically called mass spec imaging. So, in a nutshell, what it is you're able to take a microscope image, and then what they do is they take that same piece of tissue, like the next section next to it, what we call serial section. So when you make a cut, you have one section, then the very next section next to it, a few microns away, they put on like a mass spec matrix where they can do mass spec. So what they'll do is they'll take the picture like this, and here they're able to generate all this mass spec data, like showing what's in any given pixel here, like what the composition is. Then basically what you do is you put these two together, and you're not only able to show the picture, but you're able to say like, hey, okay, so my protein weighs, 12,186.4. So you can use the computer then to look at everywhere where 12,186.4 is on a given piece of tissue. So you can determine, without using antibodies, without using staining, where your protein or whatever your molecular weight molecule is of interest. You're able to actually look at it and determine its distribution on a given piece of tissue. It is powerful stuff. I don't understand quite why it hasn't caught on more, because when I went to this talk a few years ago, we were all just like blown away how amazing it was. And it's you see it here and there, but I thought it would go really big mainstream. But sometimes really good ideas take a while to catch on. So I would not be surprised if this doesn't just catch fire here in the next few years. Maybe a couple technical improvements, and it'll be a really, really big thing. Okay, so a little bit more about mass spec imaging. But it's something that, if you're interested in something like that, look it up. It's cool. Okay, now, this one we do a case. It's called intravital microscopy or intravital imaging. So what it is we're going to use microscopes today, and I said we look at slides or we look at tissue. But what this is using microscopes on a live mouse. Okay, I guess you could use it on different animals too, but I really haven't seen that. And we have a lab by us, Dr. Wong, that actually uses this. And so basically what you're doing, it involves mouse surgery to be able to place these little windows on a mouse, and then actually coming in with a multi-photon microscope and being able to take images in here. So you can look at like tumors live in a mouse actually, or depending on what you're doing. Like he looks at a lot of blood flow models, like how blood flows in a mouse and stuff. So they're actually able to open up a mouse live. Now this has become a really hot job. I've seen this advertised a lot because you have to be very good at mouse surgery, which is not easy, and then you have to be very good at microscopy. Again, especially in this kind of setup, is hard. So if you end up developing this kind of technique, become really good at it, there are jobs all over the country right now, at least in America I've seen. Even really all over the world, I've seen quite a few jobs offered to this because it's such a tricky skill set to be very good at mouse surgery and microscopy. Okay, but it's a very cool technique, especially looking at tumors and things like that, answering a lot of scientific questions. Okay, so here's a typical setup for that. Like they'll actually have like the microscope objective, the mouse is anesthetized, and they'll be able to open it up and put the objective right into the mouse and look at what they're after. So again, very technical, very tricky, but if you're good at it, there's a job waiting for you, no doubt about it. A lot of jobs, because it is not easy. I wouldn't even know where to start to do this. But there's a lab down from me. The one guy, Jay, I went and talked to him. He's like, well, yeah, I got a lot of job offers from a lot of companies. And I said, I bet, because you are really good at this. So, but he's got a family. He doesn't want to move, but we'll see what happens. Okay, so this was actually from their lab, Jay, at the Alex Wong lab. And this was looking at, like they basically injected cells into the mouse. Whoops. So, how do we get it to play? We have to click on it. See, I told you PowerPoint's not my thing. Okay. No. What? I think it's there. I'm pretty sure. No, because I thought I had it there. Because I remember playing it before. Hold on. Okay, let's go back. Here we go. Here we go. It's not there. Okay. Alright. But anyway, what it was, you see cells going through blood, basically. Okay. I apologize for that technical glitch. But when I set it up, it worked. Alright. Okay, so, just to review some of what we just talked about. What are you trying to accomplish? What is your question? Science always starts with a question. Your hypothesis, whatever. What is the question? Okay, so think about that. Obviously the image quality of the last video stunk, right? It was bad. It was bad. It wasn't very good. Anyway, it was sort of grainy. A lot like that picture this morning when he showed the Golgi, but like a bunch of squares moving. Okay, so the idea here is basically to tell you it's not about a pretty picture always. It's if you're measuring things, like he was measuring calcium. I mean, the pictures were not, I wouldn't call them pretty, but they were data, just full of data. I mean, sometimes it's about data. Sometimes it is about the pretty picture. But a lot of times scientific pictures aren't pretty. They're just full of data. Okay? It's all about data. This one probably won't work either. But we're going to try anyway. No. Okay. I bet. Okay. So, something else that's a hot technique. We have a whole setup at Case Western Reserve in our basement and different buildings. Was that there when you were there? The small animal imaging? You did some of that? Okay. You using luciferase? You did the 2-photon with Alex? Wow. And you did this with who? Chris Flask? Okay, you can show that. Okay. So Shreeja's done all this, so he's more the man on this than me. I've heard about it and talked to some people who did it. But anyway, I get a lot of slides off this because I work with the brain tumor lab, and we do a lot of this. Like they do the small animal imaging. So what it is, it's very expensive because the reagents are very expensive and the machine use is very expensive. But if it answers your question, it's worth it. Okay. So a lot of times what they'll do is like here, this was like GFP, like they injected tumors with the GFP tag. Like a lot of times we'll do mouse work. We'll inject human cells, like a human tumor actually, into a mouse and grow the tumor, a human tumor in a mouse as the system. And it's tagged with the fluorescent protein, and then they're able to look at it in these, it's almost like a, what would you say, like a box. Huh, Shrija? Sort of looks like a box, the whole thing. You put the mouse in the box, and then you image inside this box where it's really dark. Okay? But you get these kind of images, you can actually see the tumor progress inside the animal, but you don't have to cut the animal open to see it. That's the trick. You can leave the system alone, let what happens, and not disturb it by cutting it open and killing the animal and putting it on slides and such. This way you can look at it live. So the nice thing about this kind of stuff is you can follow it over time. You can bring the mouse over today, and then in a week and another week, you give a drug. Does it help? Does it make it smaller? Okay, very useful technique. Okay, and a lot of times they use, instead of GFP, they use luciferase. Anybody know what luciferase is? Right. Do you guys have fireflies here? You do? Are they like a yellow? Is it like a yellow color? Okay. We have a lot where I live. They only come out in the summer for like a month because the rest of the time it's too cold. It's too cold. But they're, so what they did is they took the protein luciferase, which is the protein that actually glows on the firefly. And you have glowing worms too, like glowworms? Like we used to have those when I lived in North Carolina. They'd glow, like you'd be on the ground, they'd be like green, like fluorescent green. It was pretty cool. But anyway, the luciferase, though, they use this protein, another one, and again they put it in the box and they use the luciferase as the reporter gene, and you're able to see where in the mouse the tumor or wherever they decide to look at falls into place. Okay, so this is all like small animal imaging kind of stuff. Okay, now this one's pretty crazy. I went to a talk a few months ago, okay, well maybe a year ago, okay, and this guy named Carl Disroth came in, okay. Now, I've talked to some people in neuroscience, and they're like, well, I get it, but what do you do with it? I don't know either. Okay, now, has anybody here ran electrophoresis? Like run a gel on electrophoresis, okay. So, a couple people have. So, electrophoresis is, you use electricity to separate molecules, and you get bands on a gel basically, and it's all done by charge. So what this guy thought was, okay, why don't we take a brain, a mouse brain, and why don't we clean it with a detergent, like a soap, and what detergents do is they emulsify lipids, so they make the lipids soluble. And your brain actually is a lot of fat, a lot of lipid. So he used this positively charged, I don't know, positively or negatively charged detergent. and he subjected it to the brain like Soten. So what it did is it made all the lipids soluble. Then he ran on electrophoresis. So what it is here actually is here's the brain beforehand, okay? And obviously you can't read underneath it. This is the brain after. So it takes all the fat like out of the brain and it leaves all the structure of the brain, all the proteins and all the scaffolding and everything intact. It's called clarity. I mean, it is crazy looking, the stuff this guy brought, okay? So take a look at this when you get a chance, because I'm sure it won't work. But it's really neat how you can actually look at like into a brain. But I talk to some neuro people and they're like, okay, well, so what? And I don't know enough about neuroscience to say, well, you could use it for this or that or the other thing. So, I mean, it sounds cool, but I don't think they've quite figured out the applications for it yet. Okay. So this guy also, who was really a clever guy to come up with that, came up with this idea of optogenetics. And I've seen this, and this seems to be getting bigger and bigger. I went to the neuroscience meeting last year in Chicago, and this was definitely catching on. It was a pretty hot technique. So what it is if you read it, is you can use light to turn cells on and off in living tissue. So you could take a neural network and you could hit it with light and it will turn on a certain set of neurons or another set of neurons, okay? And it does this all in living animals and it does it all in real time. It's really wild. Okay, and again, you'll have to watch it, but it's pretty cool. And then last but not least is this idea of rainbow, where again, they've used all these GFPs and RFPs and like they talked about this morning, all these different colors. Well, they're able to clone these all into living mice, and different subsets of neurons and stuff can actually express these different color fluorescent proteins. So you can look at all these different neural circuits all at once as one kind of picture. And like I said, I got a little like 3D of it here. You can take a look at it and get a chance. It's pretty cool. And then that would be pretty much my talk about trends and what's new and what's hot in the world of optical microscopy, and a bunch of mistakes don't make, okay? Don't make those mistakes, and always ask for help. Because, you know, like I said, science is a team game. All right, questions? Any questions? Yes. Yes. Okay, so what's the significance of the intravital? Okay, it's live in an animal, whereas everything else we've talked about at this point, looking at cells is in vitro, looking at tissue, like on a slide, is in vitro. This is like live in the animal. So you can add drugs to the animal and actually watch what happens in a live living animal. So that's the real thing about that technique that's really different, I guess. But you've got to be good at mouse surgery to start with just to get it to work. Because you've got to be able to keep the mouse alive and all this. So does that answer your question? Okay. Anybody else? Questions? Yes? I think it has a lot to do with, now you might have a better idea on this than me, like how deep you can go in intravitreal imaging. Okay, so you were pretty much on, now you could get down in the dermis? Okay. I mean, my understanding of what you can do, I mean, I guess if you open up the mouse more, you can go deeper, but in general, like 2-photon imaging it uses, you can go usually a couple millimeters deep with that kind of technique. And we'll talk more about that when we talk about 2-photon imaging, but maybe even a couple millimeters, which is, you know, it might not sound like much, but in terms of microscopy, it's huge, huge amount. So, probably a couple millimeters, I would think. Anybody else? Yeah? Okay, so we on the list. I'm going to give you a little introduction to this, you know, the next day or two. So we'll download this. Okay. Yeah, I'll accept this. So, has anybody here ever heard of a thing called ImageJ? Have you heard of ImageJ? Okay. ImageJ is free software from the American National Institutes of Health. Okay, it's image analysis software. You'll see it in papers, people will cite it, and they'll use it. And the best thing about ImageJ is, guess what? It's free. Okay, so let me see what downloads work does. So, do you want to open or save? I want to save. See, this is good. Most of the people in this room are much younger than me, and you guys grew up with computers where I started working with computers, obviously, when I was much older. First time I saw a computer, I was in junior high school. And in America, that would make me like 17 years old. So that was the first time I saw a computer. And we like made it write our initials or something. We were all like, wow, this is really cool. Okay, so it's downloaded. Okay, so we'll talk about something else when it's downloaded. Okay, so ImageJ is free software, right? And the people who started ImageJ years ago said, well, wait a second, we want to make some money. So they became Metamorph. Okay, so Metamorph is sort of an outgrowth of ImageJ. So Metamorph's been around a long time, and like I said, it's very good software. It's not the user-friendliest, but it does a lot. You can use it to run microscopes, you can use it to do image analysis, a lot of different things. And we're just going to use a little bit of image analysis just to show you how. And I'm just glad I was able to get some software off them to be able to do this. Okay. So let me pass this thing around here. I don't think you'd be able to see it well, but come on. Okay, has anybody ever heard of a slide micrometer? Okay. So basically what a slide micrometer is this, and we'll pass it around. And you'll be able to see if there's a circle, and inside the circle is a little line. Okay? And what it is, more or less, is it's a ruler. Okay? It's a ruler made for a microscope. So anytime I would sell somebody a microscope or a system, I would calibrate the whole system. Now, the thing to think about is, so let me know when it's done downloading, okay? It's going pretty steady. Okay. So I would go out to their place where I sold it, I would calibrate their system. So I would take my micrometer, I'd take a picture of 10, 20, 40, 60, whatever objectives they had, and I would calibrate each objective. So when they took a picture, they could actually get true measurements, okay? Because if not, then you're just measuring pixels. So I was able to do that for them, which was good. But, okay, so let's see. So we were able to calibrate things. And like I said, it's important if you want to know the size of what is on your image. But the big thing about calibrating the system is, if you have a 40x Olympus microscope hooked up to a certain camera, and you have another 40x objective on an Olympus microscope hooked up to a different camera, the calibrations are different. Calibrations are specific to that system. So you can't just say, oh, I took this on this microscope at 40x, so therefore it's the same as this on 40x. Okay, and the reason I talked a little bit about today about knowing the sizes to make sure they're right is we'll do scale bars. Like, for whatever reason, all the journals now want scale bars. It used to not be that way. But I think the reason they do it is there for a while with Photoshop. Have you guys used Photoshop a little bit? Okay, it's so easy to manipulate images now. It's so easy to manipulate images now that I've actually had requests from journals for the raw pictures, okay, the raw images, because I guess, there's been a lot of forgery or whatever you want to call it over the years. So my advice to you would be always save your originals, then you can always go back and do your calibrations off it and things like that. But the problem is like I said before, people bring me scale bars on things on PowerPoint or Photoshop, and I don't know what you did there. I don't know if you made it bigger, I don't know if you made it smaller. I mean, I really can't help you at that point. So just like keeping a good notebook for science, keep track of your images. The other nice thing about using imaging software, that's going pretty quick, huh? But anyway, the other nice thing about using imaging software is it's not only the image, it actually gets, it comes along with an image header. And an image header, we'll see, hopefully when we get the software working, tells you when the image was taken, tells you what the calibration was. tells you a lot of information about it. A lot of times it can read all the electronic information on the microscope you use. So, so it becomes a system. So what I have at my place, I have Metamorph on all my acquisition systems, all my cameras, all my microscopes, and then I'm able to give, like here, I'm able to give out these keys, except we have it now, all electronically done all on a network. So I have like maybe 70 or 80 people that have access to Metamorph, and I can do 5 licenses at a time. But that's our image analysis software that we use. And it's pretty cool. So hopefully we'll get it to work. Okay. Okay, there's other softwares too. There's a couple other companies. Anybody ever heard of Velocity? Velocity software? I don't know if it's over here or what, but it's another one I use. I really like it for 3D rendering, like on Confocal. Like we'll do 3D things on the Confocal, and I like it for looking at the 3D visualization of Velocity. The other things on there I think aren't very good, but that's just my opinion. There's another company called Image Pro. Ever heard of Image Pro? Is that over here? Image Pro? Anybody? Image Pro? Okay, that's another one. But I think your best bet would be, once I go, would be to download Image J. And then at least you'd have some software to start with. But Metamorph here will have the whole, I guess, the whole toolbox of things we can do. Where in Image J, you have to download what they call like little modules. So if you want to do some kind of length, you're going to have to download a module or some kind of thing. But people are always writing like free software updates to Image J. It's a little more cumbersome, but it's more or less what you're going to see here. It's just the setup's a little different. I don't work with Image J very often, very rarely, because I have this. But I think, in terms of saving money and it being free, I mean, you can't go wrong. Okay. So once you see image analysis and what it can do, you'll be like, oh, okay, maybe that's very useful to me. Okay, so another thing, we're going backwards. Okay, another thing that, it's stealing my thunder, I have the software. Okay, so the main thing with doing image analysis is this. So if you're doing an experiment, right, and your professor says, hey, it'd be really nice if this protein increased when we added this drug or whatever. So if you're sitting there at the desk and you start looking at these images one by one, I mean, you know what the experiment was, you know what samples what. So it's human nature to say, well, I think that's a little bit brighter. I think there's a little bit more. I think, you're a little bit biased. You can't help it, okay? You can't help it want it to work the way you do it. So the nice thing about using image analysis, using a computer to analyze the images, it takes the bias right out of it. So it becomes very just math, just pure math oriented, okay? So since it's pure math oriented, it takes that bias out of it. The other nice thing about it is when you do an experiment, now, I don't know how many of you work in a lab right at the moment, but I had a boss before. I would do an experiment, and I have a very exciting result. Very exciting. And I'd come up to him, and I'd say, hey. And he'd look at me, and the first thing he'd always say was, how many times did you do this? And I'd say, well, I just did it once today. He goes, don't even talk to me. He goes, you go do it again. And you do it the third time, and you get the same result. In triplicate, then come talk to me, okay? So the one nice thing about image analysis, too, is you can do a whole lot of experiments, and we'll see tomorrow that you can automate the analysis. So you can take thousands and thousands and thousands and thousands of pictures, and instead of sitting there and measuring intensity one by one by one, I'll show you how to write like these macros where you can analyze all 1,000 images at once. Okay, we'll just run through them and do the analysis. So you can get that N, that number, that experimental number way up, and make your results that much more powerful. Okay, so the image analysis has a lot of applications, okay? And then, who runs Western blots here? Anybody Western blots? Nobody? Western blots? Western blots? Western blots? Well, you use a little bit different image analysis software. You can use Metamorph, but the whole idea of a Western blot is you want to be able to measure that protein. So we can use image analysis software to measure how much is in that band, like how intense that band is. Because right now we have a system at work, it's called LightCore. It uses infrared reporters on the western blot. And therefore we can use that, and we can do intensity measurements on those bands to figure out how much protein is there. Okay, so a lot of applications for image analysis and such. Oh, we really are going backwards. Okay. What do you think, 5 o'clock? Okay, who votes? You sit here and watch this load for a while, or who votes 5 o'clock is 5 o'clock? Okay, so who votes to stay here and watch this go? Okay, who votes to say, let's call it a day and go? Okay, only 2 or 3 people vote. Okay, you have to vote. Okay, you have to vote. So, okay, I'm going to do it one more time. So, who votes to stay? Who votes to go? Okay, I think we're going. Okay, go. Oh, I get the interactive session. Okay. I'm going to do this interactive session. I look like I'm falling apart with my stuff hanging all over. Okay, so we'll let this download in the background. Okay, so the interactive session is basically me asking you questions about what we talked about today. Right? That's basically the interactive session, right? Okay. Okay. So what is everything? What did I talk about today? I said, this is everything on a microscope. The objective, there's a certain number on the objective. Numerical aperture, right, okay. This is like Jeopardy. You guys have Jeopardy in this country? Jeopardy, it's like a game show where they ask questions. It's pretty cool. I tried getting on it actually, but I wasn't able to get on it. Okay, I wrote a couple out here, so I'm cheating on what I wrote for today. Okay, so if I said I was going to do a technique of phase, DIC, and Hoffman modulation, what am I looking at probably? Am I looking at something stained? I'm looking at unstained cells. Okay, we got a winner there. Okay. Okay. Okay, so when we talked about the alignment of the microscope, what was the gentleman's name? Wow, I didn't get very far with that one. Good, good, good. Kohler, the German, right, with the little umlaut. What?
There's a prize.
There's a prize? Okay. Well, keep track of who wins the prizes. Okay. I brought some stuff from America that you've never seen before, I don't think. So I don't have it with me today, but I'll bring them in for prizes. All right. Okay, so this is multiple guess. The most important ability of a microscope objective is that it can increase A, magnification, B, what? I guess somebody was paying attention today. This is pretty good. Alright, this is pretty good. Okay, let me think what else I talked about. Okay, what's the stain called that a lot of pathologists use? Just look at something. H&E? Okay, good, good. Wow. You guys are paying good attention. Okay, so then earlier, the first talk of the day, we'll go all the way back. I didn't get this lecture, but where did the green fluorescent protein come from?
Jellyfish.
Jellyfish, right. Where am I from? Cleveland, Ohio, exactly. The home of the Cavaliers, right? World champions. Okay, who came up with the name cells? Robert Hooke. Now you probably had that before in a class, right? I'm sure you've probably heard that one yet. Right? That's like, I used to teach biology 1. That's always something I came up with. I like this part. This part's fun. Okay. I'll look back through that. I talked about. I think the jet lag's finally starting to hit me. Okay. Let's see. Oh, okay, so the thing I really didn't want to talk about, but I did, the diffraction circle. Remember, what was that called? It's got one or two names. I'll take either one of the two. It was like, remember I said I'd take a light and make that refraction pattern. It began with an A, airy disk, or point spread function. Okay? That's heavy physics. Okay. I forgot to talk about all this. Okay. So if you were going to spend money on a microscope, what would you spend the money on? Objectives, exactly. That's where I'd spend the money. And then tomorrow we'll get into the next thing I'd spend the money on would be cameras. Okay, so we'll get into that tomorrow. But the next most important component of the system is basically the camera. The microscope itself is basically just a holder. It just holds all these, it basically holds the objective and the camera in place. Okay, so if I wanted to look at plates, a type of microscope that looks at plates and slides, inverted. Now how about if I wanted to look at like a mouse eye, something bigger? Like what type of scope? Stereo microscope, exactly. What's the stand for fat that we talked about? Anybody remember that? Man, you guys are good. I don't know, you're either really good or I did a pretty good job. I don't know. Maybe a little bit of both. I gotta give myself a little credit. Okay, what's the thing if we take phase contrast of white around the cell? Very good. Which technique, Hoffman? Well, DIC or phase gives you the 3D look. DIC, yeah. Good. Okay, so if I have an antibody and I don't know if it works yet, and I want to do it the first time, am I going to do it on a cryo section or am I going to do it on a paraffin? Man, I'm impressed. If this was America, you just don't understand. Half people are asleep. Whatever. Depends what school you went to some extent, but. Okay. Okay, extension time saves. Pretty good. You guys are really paying attention. Okay, what's something, like if you're going to do prepare tissue, what's the one thing you should probably make up like very fresh? Fixatives, right? Okay, if you give me garbage, what am I going to give you back?
Garbage.
Wow. Okay, and I'm going to try to get you on this one. Okay, so the technique we talked about very briefly, but I said it would get the, like, very close to the cover slip only, really good for membrane. Wow. I'm very impressed. Okay, what was the thing I talked about? I said, when I first saw it, I said, man, I think I'm going to lose my job. Because it takes pictures, and I don't know if I really gave you the name of it. No, not that one. But the thing that takes the big picture, what do we call that? And I said you could put it in and it would just do it again and again and again and again. Right, a slide scanner is what that's called. I don't think I was real clear on what it was called. But yeah, I'll tell you, the first time I saw that, I was just like, what do you need me for? But you need somebody like me always to interpret the results, because the problem still becomes, you can take all these pictures, you can take all these images, but you need somebody with some biology experience to interpret what you're looking at. So I think I'll always be okay having a job in microscopy in that respect. Okay, so what's the technique called where we look at a mouse live in vivo? Right. Intravital microscopy, or a lot of times I'll call it in vivo confocal. Okay, same thing, but technically it's called intravital imaging. And right now, if you were really good at that, I don't think you have any trouble finding a job. Okay. What's the technique called where you take the brain and make it clear? Wow. Okay. See, in America after lunch, we just tune out. It's over for the day. Obviously not here. Okay. What do you think? Good? We're good? Call it a day? All right. Well, thank you for paying attention today. I appreciate it. And tomorrow we'll get started at what time? 10? 10 o'clock. Okay. You say what you got to say.
Thank you, sir, for that wonderful lecture. I hope everyone will be joining us tomorrow. Please be here by 10 o'clock. I would also like to say that this workshop will be evaluated and you'll be provided appropriate grades by the end of the workshop. And there will also be interactive sessions and quizzes throughout this 6-day program. And based on your participation, we'll provide a grade with you'll provide a certificate with the appropriate grade. And tomorrow, please join us by 10 o'clock. We'll be having a special talk by Dr. Deepak Nair on the topic real-time nanoscale organization of postsynaptic density, how molecular dances shape synaptic signaling. And we'll have 2 other lectures by our Dr. Scott J. Howell. And
that did lesser things for like $5,000–6,000. And this is years ago, so I'm sure it all costs more now. So this will be the code. So I'm going to say, okay, give me the registration code from a disk. Okay, it's giving me it. We're going to go to next. And I think it's going to screw up here because these keys expired because I was supposed to come last month, but we all know I had visa problems. But it will do the install. And then when we go to launch it, I think it will fail. But then we're going to try this updater. That's what the technical people from the company told me to do, so we'll give it a shot. And it should update. But anyway, well, that's going on. So what do you think we can use image analysis for with microscopes? What do you think? I mean, just what do you think we can do with it? Any ideas? What do you think we can measure? Well, what do you use a ruler for? You have or you have meter sticks here, I guess, right? So what do you use that for? To measure length. Okay, length's an easy one. So in metamorph, eventually we just draw lines and measure length. So if you measure length, if you draw a circle or an odd shape, what are we measuring then? We are measuring, right? But like inside the curve, we would call that what? Area. Right, we can measure area. Okay, but like we saw today with that first talk, what he was measuring there, he was measuring intensity, fluorescence intensity. So this is going to work out real nice tomorrow because I'm going to talk about what digital imaging is, sort of how it works, how a digital camera works, and then it'll make more sense to talk about intensity. But intensity basically is the pixel values that we'll see tomorrow, how each pixel, depending on how many photons and the energy the photons gets dumped into that pixel bucket, it generates a number, okay? Because digital imaging, deep down, is math, okay? Like I said, I don't like math. It's not my favorite thing. My son's a math major, so that helps sometimes. But okay, your linear key is expired, so this is where we'll see if we can fix this. Hopefully we can. Or I'll have to go home and write a different lecture tonight. Okay, so the linear key expired. So she told me I could run the updater. I should hear it. Okay, so meta imaging here is installed. And we're going to run the updater. When you take a picture of Okay, we'll get into pixels and what they are and such. But what is the physical size of the picture? Like I talked about a little bit this morning with the different sizes of things and such. So all the time I get people come to me and go, well, I got to put a scale bar on this so I can publish it. And I'll go, okay. Do you have the raw pictures? No. You already have it in PowerPoint or Photoshop, so you've already manipulated the image. How am I supposed to figure out how big something is? Because you've done all these different steps to it already. So I guess the one good piece of advice is always keep your original images and cool. So keep your original images and make in your notebook what magnification you took things at. So if you use Metamorph correctly, like if we were using it as the acquisition system, you can actually have like a little pull down. You can say, okay, today 20x. And all the pictures I take would automatically have a calibration applied to them that I would set up behind the scenes. I'll show you a little bit of that tomorrow. Now, the confocal here, I don't know if it's calibrated already. I'm guessing it probably is. But it's important to have sizes with your images when you take them, okay? And that's what we'll start the image analysis stuff with sizing. Probably tomorrow. So you should have put this one back up. So a lot of times here the CD will like auto load again and it'll come back up. Okay. Accept the terms again. Okay, now it says the Sentinel version is installed now, 7.42. Now there's a checkbox. Don't check that box. Okay, it's tempting you. It's saying check me, but don't. Okay. Okay, and then it makes up a folder called MM. Okay, now this is the part where it recognizes the dongle. Okay, so what we'll do is we'll plug in the dongle. If we get the USB port. We can take that one out. Okay. Okay. You can tell I'm not super technically proficient with these kind of things. I'm not real good with the phone either, but I got a 22-year-old daughter, so that takes care of the phone for me. She can handle pretty much everything. I got a 24-year-old son who's really good at computers, so I got my bases covered. Okay, so we're going to use a local key, which is this. And we're going to say read system ID from key. Now this is going to be weird because I hope this works because, well, let's just see what happens. Okay, so I've got that. Can I put another dongle in? Huh? But I have to take this out. This is the Wi-Fi, right? So I'm going to go through here. Okay, so I'm going to install the Sentinel security component. Okay, so I'm going to give you that dongle, but at this step, you want to make sure that dongle is not in yet, okay? So that's what this is all talking about here. Okay, it'll start doing its thing. Okay, so while it's doing its thing, let's talk a little bit more about image analysis. Okay, and I think earlier somebody had mentioned, I think one of the inaugural speakers had mentioned image analysis in terms of a barcode, right? Like a barcode really is image analysis. It's looking at that image and being able to analyze and extract information from there. So what's another kind of image analysis you can think of that's out there today? What else? I mean, what would be another thing that we look at? It would be a type of image analysis. Anybody? Well, what do they do for criminal stuff? What do you do in India to find out if somebody did something as a criminal? Like fingerprints, right? So fingerprints, there's a database of fingerprints, at least in the US, I know there is, and they do image analysis across those to compare fingerprints. So that'd be one kind of image analysis, right? So what we're going to do though is we're going to talk about image analysis in terms of microscopy. So when I used to sell microscopes, I was an exclusive dealer for a software called Metamorph imaging software. And at the time, Metamorph was without a doubt the best software there was for image analysis in terms of microscopy. They were the leader at the time. I wouldn't say they're the leader now, but it's very powerful software, it's very good software, and I have a lot of friends there. So they were able to give me 10 dongles like this. So what we're going to do here now is I'm going to load the software. I'm going to show you how to do it, and then tomorrow I'll pass this CD around. So we need to break up into some kind of groups. I don't exactly know how Sreesh wants to do that because I'm not sure how many people we have in the class. But I've got 9 other dongles we can use, so we can break up into 9 other groups, and you guys can install this software on one of your laptops, and then we can work some stuff interactively. You can actually do some measuring and things like that, so you're able to actually work with it, okay? So that's the plan. So what I'm going to do is I'm going to go through this or at least try to install this software. Now hopefully it'll work smoothly. We'll see. Okay. I've installed this stuff hundreds and hundreds of times, so hopefully it'll go well. The problem I ran into yesterday when I installed it was that it had expired because I was supposed to do the course in January. So they give you the dongle. You know, this is like $20,000 worth of software here. And obviously they're not just handing it out, they're a company, so they have it in their code where it will expire. Okay, so we can keep it, but it's not gonna be any good if you do that for a done. So anyway. But I gotta take it back to the US, give it back to stay on their good side. Okay, so we'll start with this install. Okay, let's see if we can get this to work.